An action potential begins when sodium flows from a high concentration down channels into a lower concentration in a nerve cell. This is called depolarization. Potassium then exits a cell from a high concentration through channels into the outside environment called hyperpolarization. The wave travels down the axon, causing a nerve impulse. Sodium-potassium pumps then restore the original concentration during the refractory period. When this reaches the end bulb, it causes calcium to flow into the nerve end bulb. This influx of calcium causes vesicles containing neurotransmitters to migrate to the membrane at the neuromuscular junction. These vesicles then fuse with the membrane, releasing neurotransmitters, in this case acetylcholine, into the synaptic cleft. ACH then binds with receptor proteins on the motor end plate, which causes it to depolarize. As acetylcholine diffuses across the synaptic cleft at the neuromuscular junction, depolarization of the motor end plate causes an action potential to propagate across the sarcolemma and down the transverse tubules. This action potential triggers calcium release from the sarcoplasmic reticulum and causes diffusion into the sarcoplasm.
At this point in the cycle, the myosin binding sites on the actin filament are blocked by tropomyosin. As calcium enters the sarcoplasm, after being stimulated from the action potential, it binds to troponin, causing tropomyosin to change its shape. This then exposes the myosin binding sites on the actin. At this point, the myosin head is attached to an ATP molecule and is in its low energy configuration. The myosin head then hydrolyzes ATP to ADP and phosphate, causing it to change shape and bind to the actin molecule. This change of shape forms a cross bridge. Then, after the ATP and phosphate have been released, myosin shifts, pulling the actin filament toward the center of the sarcomere, the process of contraction. During relaxation, a new ATP molecule is attached and the myosin head returns to its original state. The tropomyosin also changes shape, re-blocking the myosin binding sites. This process is explained more in depth momentarily.
Tropomyosin's original position is restored as calcium ions break off of troponin. They then move toward the channels on the sarcoplasmic reticulum and are transported back in. Near the membrane, acetylcholinesterase breaks down ACH and the muscle fiber is no longer stimulated and contracts no more. Collectively, the myosin and actin filaments make up the sarcomere. As the myosin heads go through their contracting process, they pull the actin filaments together. This causes the sarcomere to shorten and the muscle to contract. As the myosin heads release their ADP and phosphate and bind to another ATP molecule, breaking their bonds with actin, They return to their low energy configuration, and the actin fibers extend back outward, relaxing the muscle.